continuing with the history of neuroscience, in the 19th century, as we mentioned in the previous lecture, uh, we were just beginning to view the possibility that nerves act as wires, uh, sending electrical signals along their extent, and that's how the brain can communicate with the rest of the body through these nerves. And this was thanks to, in part, to Galvani and Bois-Reymond that we understood that nerves could conduct electricity. Now, something else quite interesting that was going on in the history of neuroscience at this time, that is early to mid 19th century, we had Gall come along, and I'm sure you've heard of Gall before in some previous psychology class, like intro psychology, presumably. And Gall is the one who invented phrenology. He was one of the first people to come out and say, I think specific functions in the brain are localized, that certain personality traits can be ascribed to certain areas of the brain. And this was a somewhat revolutionary idea because, again, it wasn't obvious that different parts of the brain should perform different functions. Gall was one of the first people to say so. And it's tempting to applaud Gall at that point, but the problem is Gall was just frankly not a scientist. He first of all just came up with kind of some strange ideas of what kind of functions should be localized. I mean, if you look over here at this map on the right side of the screen, we see how he believed different kinds of essentially personality traits were distributed across the brain. For instance, traits like acquisitiveness and mirthfulness and veneration and inhabitiveness. Inhabitiveness? What is that? I don't know. I can't even find a definition of it. So I don't even know what inhabitiveness is. And I suspect even if I did know what it was, I would have a hard time thinking it was something that was localized to a specific area of the brain. Acquisitiveness, you just like to acquire things, so perhaps this is materialism. I see no evidence whatsoever that a trait like materialism is localized to a specific part of the brain. Okay? So, to begin with, Gall's ideas of what kind of human traits could be localized was rather curious. And but fine. So you think we're inhabitive? We have an inhabitive module in our brain. Well, that's fine. Let's test it. Let's do some science. Let's go and derive a hypothesis from your theory, test that hypothesis, and see whether it passes the test. And Gall never did any of that. He never bothered really with the science side of things. And so he just simply wasn't a scientist, and this made others skeptical. So skeptical were these other scientists at Gall's idea of localizationism, again because Gall was not testing anything scientifically, that eventually a backlash occurred. It's come to be known as the equipotentiality backlash. This was a school of thought in the 19th century that was anti-localization of function. That is, that you can't localize specific functions to specific parts of the brain. Indeed, their kind of credo here is that all parts of the brain have equal potential to perform any function. All parts of the brain have equal potential to perform any function. So the frontal lobes, sure, maybe they're involved in planning and decision-making and higher-order thought, but no more so than, say, the occipital lobes or the thalamus or the cerebellum or the brainstem, that all of those parts of the brain had equal potential to perform any function. That is equipotentiality. And the curious thing is it really came in reaction to Gall and phrenology. It's often considered a knee-jerk reaction. That is that, okay, yeah, you're right, Gall had some screwy ideas, but that doesn't make the idea of localization of function completely bunk. In fact, we certainly know today that quite a few functions can be localized. Certainly the sensory functions, like your vision, is in the back of the brain, in the occipital lobe, and your audition, your hearing, is in your temporal lobe, and so forth. And there's a motor cortex, and even certain kinds of decision-making processes can be localized to specific regions of the brain. So the problem with the equipotentialists is that they threw the baby out with the bathwater. Just because Gall needed to be exiled didn't mean that the idea of localization of function needed to be exiled. But that's kind of what happened, and the equipotentiality folks kind of dominated neuroscience in the early part of the 19th century. But fortunately, we had Paul Broca come along, and he's going to pick up and run with the flag of the localizationists. Paul Broca was a French neurologist in the 19th century, and he had a variety of patients. One day, though, he had a patient come in who couldn't really say anything. All he could say was Tintin. And so Broca took to calling this patient Tintin, because that's all he could say. Couldn't even say his name. And so Broca was really quite curious about what could be this guy's problem. Why is he unable to speak? His speech was really just sounded like he was exceptionally drunk, just a really slurred speech, and couldn't really make sense of hardly anything that the man was saying. And so Broca was quite curious about this, and he worked with the patient for a while trying to help him, but the patient never did get better. And eventually the patient died. Whether it was well, it wouldn't have anything to do with the Broca's impairment that he had, but anyway, the patient did eventually die. And so Broca decides to perform an autopsy on this brain. And he's thinking, well, this is interesting. I suspect that something went wrong with his brain, is what Paul Broca was thinking. And that's what gave rise to the man's speech impediment. So, now that he's dead, let's take a look at his brain and see what we can find. And interestingly, Broca found a very sharply delineated, acute area of damage in the inferior frontal lobes. Here's the front of the brain to the left and the back of the brain to the right. So if you look where this dark area is that I've circled, you find, well, just a big hole there. And Broca obviously saw that big hole and was absolutely stunned. There was a very localized area of damage. The rest of the brain was fine. The rest of the brain looked just peachy keen, but there was this one specific region that was damaged. And his patient, Tintin, had one specific behavioral impairment. That was a speech impediment. So this caused Broca to think that he had found a language area, speech area of the brain, that a function like speech and language could be localized to a specific area of the brain. Now, this one patient alone was not enough to conclude that. You'd really like to replicate these findings and see it in multiple patients. And indeed, Broca did so. He went looking for other patients with this kind of speech impediment, and then later, after their death, upon autopsy, would find that they'd have damage to the same region of the brain. And not only did Broca do this, but after Broca's findings got out, other neurologists started to look for the same thing, and they confirmed Broca's findings. So, voila, in one fell swoop, Paul Broca managed to put localization of function back at center stage. Equipotentialists were good scientists and had some reason to be skeptical, as any scientist should be, about localization of function, and certainly Gall's work, which was, again, not science, did not demonstrate localization of function. So it took Paul Broca and this really fascinating finding before neuroscientists started to consider again the idea that certain functions may be localized to certain parts of the brain. Now for the final lecture in Module 1, we're going to finish up History of Neuroscience. In the previous lecture, we discussed Gall and phrenology. We discussed the equipotentiality backlash to Gall and his phrenology, the backlash to a localization of function mentality, and then we examined how Paul Broca swooped in and saved the day for the localization of function camp. So those were very important theoretical contributions. I'd like to now discuss a more technical methodological contribution to neuroscience made by Camillo Golgi. This is an Italian gentleman who was somewhat of a chemist. He was working with different chemicals, and he had discovered this particular kind of dye, stain. They call it the Golgi stain. And the reason he invented this particular staining process was because he was trying to get a better look at the brain. He wanted to see individual cells in the brain. And you might ask, well, I mean, we had microscopy already by this time. Why was just putting a slice of brain tissue under a microscope not sufficient? Why did he want to go and stain the cells in this tissue? Well, the thing about the brain and the neurons, the brain cells, they are heavily intertwined, and they're very difficult to see because of that. It's really like a tangled mess in there, if you will. And so figuring out where one cell begins and the other ends is really a very difficult task. It's so what Golgi needed was a way of staining a single neuron so that it would stand out from the background of all the other neurons, so that this neuron with all its arms, its processes, tangled up with a bunch of other neurons, it would still stand out because of the stain that had been absorbed by that cell. So he set about developing a stain, and sure enough, he succeeded. And for this stain, he won the Nobel Prize in 1906. This was an incredibly significant contribution because for the first time in the history of humanity, we not only have the microscopic power to look at something as small as a brain cell, but we now have a staining capability that allows us to see the structure of an individual cell in detail. And this was just incredibly useful for neuroscience, and Golgi staining is still used to this day. Now, in terms of what Golgi did with the staining procedure himself, he wasn't quite as good on the theoretical side as some other people were. Lots of people have done extremely profound work with Golgi staining procedures, just that he himself didn't do that much of use. In fact, he believed that the brain cells that he was looking at were actually just water pipes, that it wasn't conducting electrical signals, that it was conducting water, essentially. The first person to really do something significant with Golgi's staining procedure was Santiago Ramon y Cajal. We see an image over here of a Golgi stained neuron, and we can see it in great detail. Now, what you don't see here is that there are 10,000 other neurons surrounding this one dark neuron. We don't see them, they fade into the background. Only the neuron that has taken up the stain is actually showing itself. Cajal was the first person to take this staining procedure and draw detailed drawings of these neurons. He was a very good artist. He was able to draw these neurons in great detail. And he did so. And he, by virtue of his studies of these neurons, came up with the neuron doctrine. He theorized that the functional unit of the nervous system was the neuron, the brain cell. And this idea came to him after using Golgi's staining procedure to look at extremely great detail at the structure of the neuron. Great. So our last slide here is going to bring us up to modern times, to neuroscience today. Now, you may notice I stopped there at the early 19th century and haven't covered any of the history of the majority of the 20th century. And the only reason for that is because that's pretty much going to comprise this course. That much of the information that you will be learning in this course will be information that neuroscience has uncovered over the last 100 years. So rather than giving you a history lesson on it, we'll just make the course a history lesson on that. So, but I do want to tell you what the current state of the field is today. That is, what are the different sub-disciplines within neuroscience. And the first of these, what we're actually going to do is go through levels of analysis. We're going to start at the microscopic level, or even the molecular level, and we're going to end at the whole brain level. Okay, so down here at the molecular end, we have molecular neuroscientists, and they are just simply interested in what kind of molecules are important in the brain. for instance, what kind of neurotransmitters are important. Molecular neuroscientists are really here at the microscopic chemical scale of things. They're really interested in the chemistry of the brain. Now, we can go up to the next level where we're talking about cellular neuroscience. And neuroscientists at this level are sticking electrodes inside individual neurons. So it's not so much that they're studying the molecules inside the neuron, they're studying the neuron as a whole and recording its activity. And this makes them cellular neuroscientists. Now we can go up a level from there to systems neuroscience. And now we're not just looking at the single cell, we're looking at tens of thousands of cells in a single area of the brain, all working together to form a system. Hence systems neuroscience. An example of this would be the visual system. I've already mentioned that vision is primarily located in the back of the brain in the occipital lobe, and so if you were systems level neuroscientist, then you might be studying tens of thousands or maybe even millions of cells in the occipital region. Now we can go up a level from there and have what people generally call behavioral neuroscience. And at this point, you're not looking at a single system, you're looking at how multiple systems interact. And this is an invasive area of science. That is, that behavioral neuroscientists are usually recording brain activity in non-human animals, generally cutting open the head and recording from electrodes in the living brain. And these behavioral neuroscientists can study all kinds of interesting phenomena like memory and attention and emotions and decisions and navigation and appetitive behaviors, just anything you want that a non-human does, you could study as a behavioral neuroscientist. Now, we're going to go, instead of taking another step vertically in this hierarchy, we're going to take a step laterally. That is, cognitive neuroscience, I would not argue was a level of analysis higher than behavioral neuroscience. Honestly, the difference between behavioral and cognitive neuroscience is simply the subject matter. Do you use invasive measures to study non-human animal brains? Then you're a behavioral neuroscientist. Or do you use non-invasive procedures to study human brains in action, in which case you're a cognitive neuroscientist. So you can, both behavioral neuroscientists and cognitive neuroscientists could be looking at the same phenomenon. For instance, say episodic memory. There are lots of people doing research with non-invasive brain imaging techniques in human beings looking at episodic memory. And there are also lots of people doing similar kinds of study in, say, monkeys and rats and so forth. And the only difference is, in the monkeys and rats, the scientists feel a little bit ethically more justified in cutting the person's head, the animal's head open, rather than cutting a human individual's head open. So, so that's the real difference between those two levels. And what we have overall here is a hierarchy of different kinds of neuroscience. Just so you know what page your professor's on, I find myself at the cognitive neuroscience side of things. I use electroencephalography, EEG, to measure brain activity in subjects in my lab, and my interest is specifically in something called mirror neurons, which are important for processes like empathy and social interaction. So I would consider myself a cognitive neuroscientist. We have other people in the NAU psychology department who are more at the behavioral neuroscience level, and even some at something I haven't listed here, the clinical neuroscience side of things. It's really a multi-faceted level area of study, and I highly encourage everyone to get interested in it. I hope this course will help you get interested in it, and we'll learn about really pretty much all of these different levels of analysis as we go through the semester. But again, with my background being cognitive neuroscience, what you will receive from me is a class that is more heavy on the cognitive neuroscience than if you were to take it perhaps from one of our other professors. So again, that's the end of Module 1. Please proceed when you're ready to Module 2.